So welcome everyone. Welcome to Strait Science. Strait Science is an evening seminar series put on by University of Alaska Fairbanks Northwest Campus and University of Alaska Fairbanks Alaska Sea Grant. Both Northwest Campus and Alaska Sea Grant, we are public servants of the Bering Strait region. We serve all peoples of the Bering Strait region, which is the homeland and waters of the Inupiaq, St. Lawrence Island Yupik, and Central Yupik peoples. So tonight, boys, Strait Science, we can't really do any better than this because we've got a Katrin and Iken and Lee Cooper. Katrin Iken is a marine biology professor at the College of Fisheries and Ocean Sciences in Fairbanks. And Lee Cooper is he's a professor. I was going to say whatever, that's something else. But anyway, he's a professor. Sorry, he's a professor with the University of Maryland's Center for Environmental Science. And he's part of the Chesapeake Biological Laboratory that is having its 100th anniversary this year. So kudos to the Chesapeake Biological Lab. And Lee, I don't think you've been with them the whole time.
Most of that time, yes.
But most of that time. No, anyway, so welcome to Strait Science. And like I say, we don't often get a Strait Science that's being beamed from the Bering Strait very often. So right now, Katrin and Lee Cooper are on SIKULIAQ, and that's the UNOL's research vessel that is they're currently off Port Clarence, sort of southeast of Little Diomede, and headed for Nome. We should see them if you're on from the region. We should see them at the dock tomorrow morning, and then they disappear and head for their Labor Day weekend. And SIKULIAQ will turn and head for home base in Seward and start another cruise, I believe. So with that, they have been out at sea up north on the Arctic Collaborative Ecosystem Cruise, and with a name like that, you know it has to involve a lot of teamwork, a lot of coordination, just like everything else in the Bering Strait region. Teamwork makes the dream work. I'll leave it there, and we'll let Katrin and Lee take us on board SIKULIAQ and let us hear what you guys have been finding around the region and up north. Thank you so much for coming.
Yeah, thank you, Gay, for the introduction and for having us. So as Gay said, we are live from the SIKULIAQ, and want to tell you a little bit about the things that we've been doing and what we've been up to here for the last 2.5, 3 weeks. First, a little bit about what this cruise is about. We had a cruise from August 11th to the 29th, and starting out with thanking our funders, and to see them on the right here with all their logos. And I want to provide a specific shout out to Dave Allen, the program director from NOAA, without whom and his tireless work to make this cruise happen, we wouldn't be here today. So what are we doing here? Our major research questions are sort of what the ecosystem changes are occurring here in the Northern Bering Sea and the Chukchi Seas, and then how environmental conditions and potential changes thereof influence marine organisms. And so we do a lot of work here to look at the ecosystem status, from physics to the biology, looking at biodiversity, and oftentimes try to create time series so that we can really follow along what these changes are. A large portion of our work too is to retrieve and deploy moorings, and we'll talk about that a little bit more in a moment. It says collaborative ecosystem cruise, and it's collaborative because we are together from at least 4 programs. That is the Arctic Marine Biodiversity Observing Network, AMBON, the DBO Distributed Biological Observatory, the CEO Chukchi Ecosystem Observatory, and then EcoFoci, Ecosystems and Fisheries Oceanography Coordinated Investigations. First of all, this is the science team, a very happy team, and did awesome work and worked really well together. It was a pleasure to have everybody on board. But we would be remiss if we wouldn't thank the absolutely amazing crew of the SIKULIAQ. Without you guys, nothing would work. We wouldn't be here and we wouldn't be able to do our work. So Gabe asked about the conditions, and so this is just a very broad overview here of the conditions during our cruise. You see here, each dot is a station that we occupied and measured various environmental conditions. On the left, you see sea surface temperature, and on the right, sea surface salinity. On the left here, and the warmer red colors with warmer temperatures, and you can see here warmer temperatures in the south, and it got colder to the north. That's really not that surprising. We do see the warmer coastal waters. That's the Alaska coastal current, which is always a little warmer. But it really got colder, and particularly if you pay attention to surface salinity, it got a lot fresher. So these colder colors here are fresher water. And that is because we actually ran into ice, and the surface had probably a fair amount of sea ice melt. And so that caused some of the fresh water there. This is an image of the ship track, and the track is, the colors are also sea surface temperature, salinity, excuse me, and the black, little unfortunate color choice is low salinity. And you can see here, this is ice. This is C9. This was a mooring site that was in pretty good ice cover. And so you can see on this ice image here, ice, and then also scattered ice in some of the other regions. And this is what it looked like at C9 up north, where we were trying to deploy a mooring, which was a little challenging.
Katrin, for a lot of people don't, oh, maybe you're going to talk about what a mooring is?
Yes.
People don't know often what a mooring is. It's,
yes,
so thank you very much.
I should have said that earlier, but I was going to explain it here. So we did mooring operations. A mooring is an instrument that is deployed in the ocean. It's tethered to the bottom, typically by an anchor, such as this train wheel that you see here, and then a long chain and rope, and then instruments in the middle, and it's held up underwater with a float. And so these instruments are deployed year-round, and they can take measurements year-round. While on the ship, we can only take measurements the moment we get there. So these moorings are affording us an incredible view into the year-round situation. So we had a number of moorings from the EcoFoci group. On the right here, you see a map outlined in green. Those are all the mooring locations that we serviced. And so quite a few, and they are always following the same pattern. First of all, the mooring that's in the water from the previous year is being released, and it comes up to the surface, and then the ship has to go sideways here, and then bring in and recover the mooring. Then the new mooring is being put together and then deployed. That sounds easier than it is, but the team has been incredible in putting all these moorings together and getting them in the water. Of particular note this year is a newer type of mooring that was brought out up here at the C2 location. It's a RISE crawler, and crawler stands for profiling and crawler. And I hope I'm getting this right, but it's an instrument that actually is able to rise up and down on that chain, basically between the anchor and the float, and take measurements. Here, temperature, salinity, dissolved oxygen, chlorophyll, and basically goes up and down and records these variables in near real time. And what you get is in these figures down here. So for each day, you can see how there is a measurement going up and down. Depth is on the y-axis here, and then it goes up and down and takes measurements here of temperature and salinity. Now, the problem is during winter when sea ice comes in, then a mooring that goes pretty much nearly all the way or all the way to the surface is very prone to being damaged by ice. And so this mooring can sink itself in the fall before the ice comes in, and then refloats itself in the spring. So that is a very cool, I think, development in this mooring operations. We also, the Ecofoci group also deployed a Trios Ramses instrument that measures hyperspectral radiometry. We called it affectionately the light thingy. And so it floats on the surface for a little bit and takes measurements, hyperspectral measurements that together with water samples taken at the same time can be used to ground truth and develop algorithms for hyperspectral platforms, such as the PACE satellite. A particular type of mooring that was also deployed and retrieved were acoustic moorings for marine mammal sounds. And so here you see a map of all the moorings that were deployed and retrieved, a total of 14 instruments recovered as well as deployed. And there are a variety of different instruments that can record marine mammal sound or any sound, and then also one instrument that is particular to record echolocation clicks to try to track porpoises. And so again, these instruments are in the water the entire year and have been in the water for many years now. And so they have a time series going from September 2012 to August 2025. As you can imagine, they record continuously every single day. And so there's a lot of data that will need to be processed later. But Dana Wright here on board gave us a sneak peek of the data. And so you can see here from these moorings, this is a humpback whale sound from July 25. This is a bowhead whale song from February. And then here you can see bowhead and bearded seals in April 25. So lots more to come from that. The UAF team also has a mooring array, the Chukchi Ecosystem Observatory, or CEO, which is a group of moorings sitting together in the northeastern Chukchi Sea. It has a lot of different instruments on it, and so it was successfully recovered, measuring of all kinds of things, temperature, salinity, of course, acoustic backscatter, currents, nitrates, and other nutrients, and some ocean acidification parameters, and irradiance, chlorophyll for phytoplankton biomass, and then also a underwater sound instrument. The UAF team also brought a little ROV. It's a remotely operated vehicle. You see it here in the yellow circle. And it came to the rescue because we had a few instruments that didn't respond in past years to come up to the surface. And so we used this ROV, or they used it to great effect. And you can see here, this is the team, Lydia and Pete and Savannah. And you can see on the screen a first visual of this little yellow float, and they were successful in hooking a line to this tripod, which has been in the water for 3 years, and bring it up. We were then also able to bring a short-term mooring that failed to surface last year, and so we had an opportunity to go there and use this little ROV again, and managed to get the hook onto this tiny little line here and bring it up. Congratulations, ROV team. And while we were on the way, there was a glider, a UAF glider that was supposed to come in about a week from now. And since we were there anyway, we picked it up. So a couple of sort of extra pickups. I'm turning it over to Lee now to talk about some CTD work.
Yeah, okay. So one of the key things that's on the ship, of course, is what we call the CTD. It stands for conductivity, temperature, and depth. Conductivity because it measures the salinity, temperature because it obviously measures the temperature. Those devices are there on the bottom of this contraption, and there's also a depth sensor, so we know what depth it's at. And then above it are all those little gray containers that are really bottles that go down open, and then they can be closed at different depths. And then the electronics, uh, we can monitor this as it's in the water, and you can see the different things that are measured as you go down in the water column, and you can see, yeah, the temperature changes, the salinity changes, lots of things happen, and so this helps us understand everything that's in the water column that can be measured electronically. Then we go on and measure things that are in the water that are closed at each depth, and everybody wants some water to take home and do something with. So this is an example. Inside that little door on the right opens. The CTD goes out that door, and then it comes back safely into this little garage for all the water sampling. And there are the people on the left waiting to go out and get their. So these are some of the things we're collecting. Inorganic nutrients, so that's the fertilizer for the plants, dissolved oxygen that tells us how much oxygen is in the water that supports life. Chlorophyll A is the how much plant life there is, and lots of other sampling that's done. Some of this has to be done on board, for instance, the dissolved oxygen, but in other cases, we're sending the samples back to the laboratory to do the analysis at a later date. And then another thing that's being done with the water samples is we're also measuring how much carbon dioxide is in the water itself. You know that it's increasing in the atmosphere. Some of that CO2, that carbon dioxide, gets into the ocean, and just like Coke with CO2 in it, bubbles in it, it actually increases the acidity of the ocean. So that has some impacts on organisms that live in the ocean. And one other thing, of course, is because the water's cold, there's more CO2 or carbon dioxide can be dissolved and absorbed into the ocean. And I won't have time to go into all the details, but carbonate, so the animals that have shells with carbonate in it, it's corrosive, the carbon dioxide. And so we have and it's a little bit of complicated chemistry, but on this chart on the right, you can see this little Greek letter omega. So when you go below 1 omega, that means if you stuck some calcium carbonate, a seashell in that, it's not thermodynamically stable. It's going to dissolve away. And so you can see we do have corrosive conditions in places, particularly at depth, in the areas that we're sampling. And so these are all the samples that collected and analyzed as part of this program. We're also keeping an eye out for harmful algal blooms. There's a couple of species that are we know now are up here, some in the form of cysts that are generating toxins that get into the food web. And so this is potentially of human health concern if those toxins get into clams that are filter feeding, the phytoplankton that are in the water. The clams get eaten by the walruses, and then occasionally people are harvesting the walruses, or anyway, it gets into the food chain. So keeping track of what's out there is important. So that's an important part of what we're doing on this cruise. And one way we're doing that is using a device that's called an IFCB. It's an imaging flow cytobot. So we have this little device that's here on the right, where I've got my mouse. Whoops, I went back. Let's go back. Thanks. The water is getting pumped out of the surface of the ocean and into this little device. You can see it says danger on it. There's a laser in there that's sensing cells and taking pictures. And so on the left, you can see all of the cells, and we're looking for a certain kind of cell that's associated with this Alexandrium. And on August 16th, we found some in the water. So there was a minor bloom. And it was northeast of Gamble on St. Lawrence Island, and you can see the little colors show up there on the mouse there. But by and large, we didn't see what we've seen in the past. So there is Alexandrium up here, but not so much that anybody need be concerned about it, at least this year. So another thing that's being collected and will be analyzed later is what we call environmental DNA or eDNA, and that's because everything that's in the ocean is shedding a little bit of DNA from the skin. There are things flowing into the ocean from rivers and so forth. So if you can filter the water and get the DNA out of it, the little fragments of DNA, and then process it, we can figure out what's been there, whether we've seen it or not. So the process includes collecting those water samples again out of those bottles on the CTD. Here's the number of samples and the places it was sampled, and then extracting that DNA, which is done back in the lab. And then there's some special enzymes, something called PCR or polymerase chain reaction, that's used to detect what base pairs, the DNA base pairs that are in the DNA that's in the water. You hook enough of those together, and you can figure out, oh, well, that's unique to this animal. And so we've got this kind of a whale, or we've got this kind of fish moving in there. And so even though we're not here all the time, we can detect things that may be there in the ocean. And so the sequencing will be done later in the lab in Seattle, and we'll consult these databases. They're also called libraries. Find out what animals identify with that. Okay, and I want to take a few minutes because very important part of what we do is not only the CTD, but what we do on the back deck, and we're out there collecting. The first thing we do is use a bongo net. We've got a picture of that. It's kind of like a couple of drums out there. They're collecting zooplankton, so little microscopic to just barely visible organisms that come up in a net, and species are looked at. Then then we get the back deck a little muddy. We'll get some sediments, and we'll look at the animals in there. On some of the stations, we used a core to get more careful sediment samples, and then we'll do a beam trawl. We'll get epibenthic fauna and fish, and there are also fish larvae that come up in the bongo net. This is what the zooplankton toe looks like as it's deployed. You can see the fish larval nets and the two big bongo nets, and these are some of the organisms that come up that are, some of them have to be looked at with the microscope to identify them. And we do or we have people on board who are taking a quick look, which is the next slide. The objective, of course, is to see this is the base of the food web. How are things doing? What's available for food? This includes things like krill and copepods that are really the real base of the food web that fish and everything going upward need, as well as what the fish larvae are that are out there that are the basis for the fish populations. And so there's a look on board the ship. There'll be a more detailed analysis done at home with the preserved samples, but we're interested what's out there as a first look. And so this is being done on the ship with microscopic work. And so there's a bunch of different organisms, the gnats, decapods or crabs, krill, that are important for the fish, and they're of different sizes, or they're important for the whales. Copepods also a key organism for feeding food. And so we've been out here enough times that we have a little bit of a record. It's not continuous, but you can see in different years their variability in how large the copepods are, and so there's some impacts on the food web in terms of understanding how things have changed over time. So the different sizes of the copepods, whether they're large or small, as well as the persistence of the krill, or also called euphausids. They're important as part of the food web, and they're particularly important for the seabirds and marine mammal diets. And then on to there's a picture of me, so I know this is very important part of the sampling. We're out there looking at what are the animals that are in the sediments, and we use this tool, very sturdy grab. That's really, we call it a Van Veen grab, but it's really a mud grab to go down to the bottom and pick up stuff. We do it enough times so we think we have a statistical understanding of what's down there. We're looking at what's in the mud. Is it clams? Is it amphipods that are important to the gray whales? And we can provide a few results because we've been doing it for a few years. Is that the clams that are prey for spectacled eiders and walruses southwest of St. Lawrence Island, that population is stable at some of the sites, but it's declining to the south. So there's some changes underway. The amphiliscate amphipods that used to be present north of St. Lawrence Island, they really have disappeared and become a much smaller area that has those amphipods. And then once you get north of the strait, there's still a lot of food up there, big Makoma clams, which are tasty to the walruses. And so there's some changes that are going on, and we're still working to understand that. We do some other things with the mud, and I don't have time to talk about all of it, but up in the upper right-hand corner is Christina and Jackie working with a core that they use for incubations to figure out how fast oxygen is disappearing from the sediments. These are data here, sediment chlorophyll. So this is live chlorophyll that's settled to the seafloor. So you can see where there's a lot of food getting to the bottom within the last few weeks to months. And you can see their differences depending on where you go. When we went to Icy Cape, there wasn't much. When we went north of Bering Strait, what we call the Chukchi hotspot there between Point Hope and the International Date Line, circles are bigger. That's because there's more sediment chlorophyll. We also do grain size. This is from a cruise earlier this summer. We'll do the same thing and see the grain size and do some analysis of the sediments. We also take a few spoonfuls of mud and give them to other people that are doing. So we've got people looking at harmful algal cysts on board. We're taking other sediments to look for a persistent lipid molecule. That's a fat molecule that's only made by ice algae. So that's going to a scientist at American University. We're doing the grain size stuff ourselves and looking at the carbon and nitrogen content. And then we have another colleague at the USGS, US Geological Survey, who's looking at ostracods. These are, I'm sure everybody wants to know what an ostracod looks like. There are many different ostracods, and they're very sensitive to water temperature and how much ice is in the water. So they're varying, and what Laura Gemery is finding is that there's some ostracods that are sneaking in here now in coastal waters that really hadn't been seen here before. They're coming from the Pacific. So it's getting warm enough that we're getting species invasion. So we're giving her a little bit of mud. She's coming up with all of these sediments. Now it's time for an interesting change of pace. We're going to talk about equifauna in the small trawls, and I'm going to let the expert on that, Katrin, take over.
Yeah, thank you. When Lee said that, it's important to talk about the mud, I mean, now we are really culminating here. And the most important thing, of course, just kidding. Epifauna is critters that live on top of the seafloor. So while Lee and Jackie and their team are looking for things that live within the sediment, me and my team here, Franz and Zach, we're looking for things that live on top of sediment, including fish. And so here we see, this is just the invertebrates. You can see biomass distribution along the various lines where we collected. And so a part can be a lot, which means we process a lot of samples. These are some pictures of the stuff that we get. So lots of different things, from brittle stars, very, very common, to very beautiful little basket stars, and many other things. And what I'm showing you here is a plot where each dot basically is the culmination of the whole sample that we collected at a particular station. And I have color-coded them by lines, so DBO 1 through 5 and the IC line, and then by year. And the closer dots are together, the more similar they are. And basically the takeaway here is that over the years from 23, 24 to 25, we get fairly consistent groupings of these samples. Here the DBO2 line, this is from the DBO3 line. And so what we see here is this is a coastal group. They are very different in the animals that we find than a lot of the rest, which this all kind of clusters together because there are brittle stars. And I'm always joking that probably the ship for the next 6 months will find brittle stars somewhere hidden on their back deck. And we also see that DBO2 is very different. And the reason for this is there are no brittle stars. One animal that we pay particular attention to are snow crabs. Probably most people know that snow crab in the Bering Sea has, the stocks have collapsed pretty dramatically. So there's also an increased interest in knowing how snow crab are doing in the Arctic, where they are not fished. They're not at a size that can be fished. But it's interesting to follow through how they're doing. And so this is the catch or the abundance or biomass that we found of the crab at different places. And you can see there are a couple of places where there were a lot of them. And then we also measured from every single crab the width of the shell here, of the carapace. And thank you to Zach for doing this. And so we can follow where are the bigger crabs and where are the smaller crabs. So other places where are little hotspots for babies. And for instance, here we can see here, this is the size and this is the frequency and these are the different lines. And we can see, for instance, DBO2 had a lot of little babies and DBO3 as well. While in DBO1, we had mostly larger crabs. So we can follow a little bit, you know, where are the big ones, where are the small ones, and of how many are there. Speaking of crabs, we actually found an adult blue king crab. You see here that was hidden in a mooring over up at 71 degrees north. That was part of the CEO mooring cluster. And you see here, Christoph Gabaldo, our captain of this cruise, holding this king crab. We have previously detected adults in the northern Bering Sea and juveniles at DBO3, but at least to my knowledge, we have not found adult blue king crab this far north. So this is a pretty interesting observation that we opportunistically really made here. Who knew that the moorings catch interesting epifauna? I mentioned we not only do invertebrates, but we also catch fish. So there's something fishy going on here. This is Franz put together a plot showing us what the composition of the fish was that we found. So for the most part, we found cods. A lot of that are Arctic cod, but also other cods, Pacific cod, both little guys as well as adults. And then snailfish, most of them are fairly small. Pricklebacks were also very common. Sculpins, also common, but actually a little less common than in previous years. We'll see that in a moment. Eelpout, poachers, and also a few flatfish. So this is a comparison of the proportion of the catch by some of these major groups that you just saw pictures of from 2015, 17, 23, 24, 25. And you can see that there's same groups occur, but the proportion changes. So in 2017, for instance, were a lot more cod than in any of the other years. And so there are a couple of changes that are happening. And then Franz looked at how much do we catch in fish. And that's actually quite interesting, same years. And so this is the CPUE, which is catch per unit effort. And so you can see here that in 2017, it was much, much higher than in 15 or in these more recent years. And this coincides with a warm year where there was very strong Pacific water influence. And so it probably brought a lot more of those Pacific species into our study region. And now we're moving up from the back deck to the bridge. Important and very interesting things happening on the bridge. We have Mari and Taylor up on the bridge. In addition to, you see here, this is Audie and Gary from the crew. Marty is looking for seabirds and Taylor is looking for marine mammals. So the marine bird surveys, I admire these guys. They are up there, rain or shine as they can, and any sea state as they can. They are up there and look for birds and marine mammals continuously, visual at sea surveys, and they look for the richness and the diversity of the species. Where are they in relation to our ship track, and what's the distribution and community structure, and then relate them later when we have all the data together to environmental conditions. And then we can also relate them to, as Lee said earlier, particularly zooplankton or larval fish in terms of the prey abundance. How do these apotrophic levels relate to those particularly zooplankton and larval fish species or environmental conditions? Couple of examples of what Mari observed for the birds. These are various auklet species, crested, least, and parakeet auklets. And you can follow here, this is our ship track in black. And then observations are in these colored dots. If there are no dots, then this could be that either we transited here at night when they were unable to observe, or we didn't see anything. So that's not necessarily discernible from this plot. But you can see here there are certain areas where there are a lot of observations, while in other areas it's fairly sparse. This is another example of northern fulmars in green, and then in orange, the short-tailed shearwater. And you can see short-tailed shearwaters are probably one of the more abundant species that we observe. And you can see they're really everywhere. And they come here to the Arctic to feed in the summer, and then they actually transit and migrate all the way down to Australia, at least among others, I'm not sure, to breed. So it's an amazing species that really depends on the rich food sources up here to do these long migrations. This was a curious observation, two snow geese that were probably migrating back from their breeding ground in Wrangel Island, and we observed them, two snow geese, 60 nautical miles southwest of Point Hope, flew by the ship. That was pretty cool. In terms of marine mammals, it's been a little sparse, but Taylor was able to see 3 species of whales, fin whales, gray whales, and killer whales. A lot of gray whales down here around the DB01 line, which is interesting because last year we did not observe them there. We observed them more up here in the Chukchi Sea, where we didn't observe them this year. Although keep in mind, again, a black line can also mean that we were transiting at night and couldn't observe. There was an observation of a killer whale around here, and then also fin whales here. And then we also pinnipeds, of course, and then here was an interesting observation of walrus that was quite unexpected to see them this far south at this time of year. And then there were a number of more observations up here, and this was when we got into the marginal ice zone, and they were hanging out on those ice floes that we saw there. That was very cool to see. And Taylor provided a couple of very nice pictures of the marine mammals that she observed. And yeah, this is sort of our highlights from this cruise. And with that, I hope I'm not butchering this too much. I'm saying, or we are saying, Kwayanak. Thank you very much.
And that was a lot of different, you got a lot going on SIKULIAQ, as always. So I guess while everyone's thinking of their questions, now is a good time to throw some, as a Straight Science audience member, throw a little love to Katrin and Lee in the chat. So this is a time to thank them for the presentation. They're taking time out. I think they're heading for Nome. They're at the end of a long trip, and we really appreciate you guys coming and giving us the what for. I know I've got a couple questions, but we'll open it up. First, I want to say there are there's two people I think that are calling in. If you guys have any questions, just blurt them out, because it's hard, like I said earlier, it's hard to be a phone call or on a Zoom call these days. Everyone forgets about the phone and they're all about the picture. So everyone else, you can raise a hand or write it in the chat if you have a specific question. So we'll give people
Thanks
a
for
minute.
the kudos that are coming
Yes,
in.
thank you. Yes.
Yeah, great job. So while they're doing that, I guess I'll ask. So we know and maybe you already did this, but your teamwork was because you had so many different groups, right? I mean, it was hard to actually keep track. Sometimes Ambon, DBO, NOAA, I don't know who's what, but is that's sort of standard, or is this a real effort to pool together and everybody pitch in?
It's an effort, obviously, but I think it's a very established group at this point, and we've done this for a few years now.
Okay, so
And
it's something not new in that regard.
no, but it obviously is continued effort, mostly from the funding perspective. We all know this isn't, easy to finance the science, the ship, and everything. And so it's more than people working on the ship together for the weeks that we're here. There's a lot of work that goes into it beforehand, and coordination, and agreements, and also understanding what the priorities are for everybody, and working together and being collegial about it and everything. So it's an ongoing effort, but it's also it's a really good working relationship.
Awesome.
Yeah.
That's always
I
good to hear.
concur with what Katrin said. We've been working together. So, some of us work for universities, some of us work for NOAA, some of us work for parts of the University of Washington that works real closely with NOAA. We're all working in this together, and we'll be talking with each other over the next months to share data that some of the things we alluded to we don't know now, but we'll know in a few months once the analyses are done.
All right. How do you recruit the team? You have a question in the chat from Lillian. Good question, Lillian. How do you recruit the team, and who pays for it?
Well, different agencies are paying for this. Like Katrin said, Dave Allen at NOAA's GOMO program was really key to sewing all of this together. But we get some money from NOAA and other agencies. All of the little symbols that Katrin showed at the beginning help put this together. So I see Danielle Dixon's on. So Alaska Ocean Observing System is part of this. And and North Pacific Research Board. Hi. Yeah, is Danielle provides that support. So there's a bunch of pieces that come together, and we're meeting the needs of, or trying to meet the needs of, all those agencies and players in the observation work.
Yeah, and so the funding for different groups. So for, let's say, the seabird team is different than the funding for the epibenthus and fish team, and is again different from the other teams. So we all come in with our funding to do the work, and then a joint platform.
So you get together
in terms of people recruiting, that's up to every team to find the people that come on the ship and do the work.
Yeah, I'd say a lot of the team members that we have, they're graduate students. That's very common as a training ground to train the next generation to come forward. So when we're when we're going out the door, or that there'll be somebody to replace us to continue some of this good work.
And Lillian, I can add that, you know, when you live in the Bering Strait region, a lot of the people on the ship are not from the region. They're coming up from large universities or, you know, big cities like Seattle and whatnot, or Boston or wherever. And so I think there's just for here, we're making inroads a little bit for the region. So we have regional students here, born and raised in the region, and we just had one of Nome's own, Nome Belts High School graduate, go on the Fish and Wildlife Service USGS Walrus cruise recently and did a fantastic job sampling for the harmful algae. So she works for the Norton Sound Health Corporation Office of Environmental Health. And that's a great example of what's a great question you have. And I think it's something that we're paying attention to in Nome, Nome Eskimo community, Norton Sound, UAF. We're all sort of, it's a great question, and we're trying to make sure that people here get an opportunity as well in the future. So thank you for asking that, and thank you, Katrin and Lee, for answering that. People wonder, how can you get on these things, that kind of thing. It's so exciting and very, you know, really important work you're doing. So.
Well, maybe also to add to this, obviously a lot of us leave the ship with a bunch of samples and not necessarily data. So, you know, some of the things that samples were taken will take a little bit of time to figure out what they tell us. But since we've been doing this for a few years, and we had a number of examples here where, you know, we're starting to establish these time series, and it really becomes obvious of how valuable that is, how you can look back in time over the last 5-10 years and see how things progress or develop, or outlier years and what happened. And so that's where the real value then also comes in, to be able to understand actual changes.
So thank you. No, any other questions? I've got a couple up my sleeve, and maybe that'll bring it on. For each of you, I guess we'll go Lee first, then Katrin, because yeah, Katrin needs a break. She's been talking nonstop there for the yeah, well done. So my question is for you, Lee, and we know that you don't have all the data back. I mean, you have the samples, but you haven't been able to do the analysis. And knowing that, it's a two-pronged question. You ready?
Okay.
What do you think is the greatest physical and biological change in our northern waters? And what was your most unexpected finding to date?
Well, I think the big change that we've all seen is the disappearance of sea ice. So that's related to warmer temperatures. So the water temperatures, as you mentioned, are high this year, at least down here in the Bering Sea, and that influence is showing up in the Chukchi Sea. So I think that physical change in the ecosystem is really driving a lot of changes that we're seeing. The most surprising change,
surprising
I think
thing
that
on your cruise.
well, you know, that king crab was really a surprise. I
Yeah,
think
that's a shock.
I think about all these people in Wainwright and Utiavik who are gonna start thinking about harvesting king crab. If that's you know, there's some changes. Well, you know, seeing these organisms come in that we haven't seen before, fish moving further north, species that are southeastern Bering Sea showing up in the northern Bering Sea, a lot of things that are biologically surprising. Those are harder to detect, but we're seeing that. So some of the hard work that people are putting in on the ship and the sampling will pay off in terms of, yeah, there are big changes happening, and they are sort of surprising.
So that
And
crab
maybe
actually,
sorry,
go ahead.
just to chime in, because Lee mentioned the king crab, and Ned has a question in the chat about, well, where on the mooring was it? It actually was on the tripod that was recovered with the ROV, and it had wedged itself between two instruments, so it wasn't even immediately visible.
Wow. So for us, just so you know, those Hanasaki blue king crab, or they're not a blue king crab. They're a Is it a Hanasaki? It looks like a Hanasaki, which is even crazier.
Okay.
All
Is it a
right.
blue king
Well,
crab or a Hanasaki?
well, we looked at it and said, "Oh, I said, and I'm not a
Mm.
crab expert. Oh, it looked like a blue king crab, but I'll stand anybody who's a
No, I
crab
don't know.
expert.
I'll
Yeah.
wait for a crab
Well,
expert.
we have it. We are taking it home so we can look in more detail.
Okay. Yeah, no, that's a shocking thing. The size of that and being so far north. So wow, interesting. Um, and then Katrin, thank you, Lee. So for Katrin, what do you think is the greatest physical and biological change in our northern waters? And what was your most unexpected thing so far? I mean, you may have all kinds
Sorry,
of
I
things
stole
that
your
you'll
thunder.
unpack later.
He stole my thunder about the blue king
Oh, okay.
crab or king crab in general.
All right.
Um, well, honestly, I have to really echo what Lee was saying, that that the changes in the physics that we see, be that temperature and sea ice and everything. And so while I showed an image or a graphic about that the composition actually is fairly stable, at least over the last 3 years, we do observe decline in how much biomass is actually on the seafloor, and so that is something we're tracking. I haven't put this year into sort of the big picture of that yet, but I'm expecting there to be sort of a continued trend of where we're seeing a decline of how much biomass in these invertebrates there is. And yeah, I echo the king crab. Sorry.
And know that that's actually, maybe that's something, Katrin, you might want to hook in with the Nome here and some of our entities that are responding to dead birds and dead marine mammals. We have seen over the years, since almost consecutively. We had a reprieve last year and a little bit this year, but the number of animals that are just starving is something that you may want to check in on that. Feel free
Yeah.
to do that.
Yeah,
I see
Gay, it looks
Charlie
like
has his hand. Yep,
we
Charlie
have a
has his
question
hand up,
too,
and
also
there's
in the chat.
also in Ken's iPhone in the chat. So Charlie,
Yeah.
hang on just one sec. I see them pop up first. From the perspective of the bird and marine mammal observers or anyone else, is ship traffic recorded? Are you guys keeping track of that? I think with your mooring, the instrumentation on your moorings, are some of that keeping track of noise of ships, or
Yes,
just
some of
looking for
them
marine?
record any kind of acoustics, any kind of noise that there is. Although I'm not having necessarily a dedicated ship traffic surveys, but there are people that are, other entities that are keeping track of ship traffic, not necessarily based on noise, but based on ships going in and out.
The AIS sort of thing. We are seeing a lot more ship traffic. We used to be up here and we were all by ourselves, and now, yeah, National Geographic cruise ship goes by and it's not that amazing. Or tankers and LNG tankers on the Russian side. So there's a huge shift in the ship traffic, which it endangers the birds and the marine mammals.
And on that, know that we've had, I don't know, Charlie will know probably more, but we've had 3 or 4 U.S. military drills. I'm sure there's Russian activity as well. So even on AIS, you won't see those vessels. The military all over the world don't turn theirs on, so they don't want you to know where they are. And so you can add in all the traffic that will not be recorded by AIS, which I can tell you we have right now. So that's also a kind of puts a wrinkle in what you think is going on and what you can see is document going on versus maybe what is going on. With that, thank you for that question. Charlie, take it away, and thanks for being so patient. Charlie, you are muted. And the button would be on the,
Okay, I got it.
got it.
My finger's
You're good.
cold or something. I don't know.
Yes, all right, you're
Anyway,
good.
now I've been doing tours for the various ships the last 3 days. But yes, the crab you saw is a Hanasaki crab.
Yeah,
You
Okay.
can tell because it's so spiny. The legs are short in comparison to a blue or red crab, and a golden king crab or Hanasaki crab look very similar, but chances are it's a Hanasaki crab because it's so shallow. That's quite a story in itself. They've been moving north as well radically. So St. Lawrence Island, Diomede now can reliably find Hanasaki crab. So
yeah,
that,
I was
Like, on
thinking,
our surveys in the
Charlie.
last 3 years, we have not found them regardless of which specific species it is. So I agree. That's still quite remarkable. Yeah.
Were
well,
you
that
far to the
that's a
west? Oh, go ahead, Charlie, sorry.
well, that's Hanasaki crab species is really making its move right now, and much more so than red. Reds in my 40-year career have colonized Kotzebue Sound, but blues have been pretty stable. But Hanasaki crab are coming on like gangbusters. So that to me that's very interesting, and I'm a former crab manager.
Okay. Well, thank you for that
So-
insight.
Yeah.
Thanks for the ID, Charlie. That makes me feel a little better. And the first Hanasaki, I believe, that was detected in the Bering Strait region was pulled out of a crab hole at Diomede in 2003. 2003. And you can see that. I think they still have it on the wall at the Fish and Game office. So, yeah. So what? We are 22 years later, and you're pulling that. That's pretty big for any kind of king crab, I would imagine. It's 71 degrees north. So, thanks, Charlie.
Yep.
All right. One other thing. Yeah, follow up on the species. Bring it on over to Fish and Game. Bring it on over when you got it when you come in tomorrow, and there's a lot of people that would like to take a look at it. I bet you Charlie would. Charlie, are you available tomorrow if they want to show you that crab?
Yes, I'm available and at home, but
Okay.
no.
All right. Well, if you want to ID it, Katrin, we can do it right here.
Sure.
The preliminarily, yeah. You got my number or text. So then the one thing that you guys mentioned was the HABs, and so that was of interest to us. And so on the way down, I haven't heard that you guys have seen anything. Your machine, your IFCB, that canister with the laser that taking pictures, magic, pure magic. That thing is operational, but it hasn't alerted that there's a problem so far on your travels south. We were a little worried about, you guys picked some of that up, and we were grateful that we were notified about that. And then we were like, whoa, we need it back. We need you back. And you came back, and you're coming back, and so far so good. That's what it sounds like.
Yeah, we did make a little stop. Now there's this area that was identified in 2013 in a paper that was published by Japanese scientists. So that's this little pitch for the international cooperation too.
Yeah.
Identifying where this bed was in near Ledyard Bay. So we made a stop there and took a few Van Veen grabs just for HABs in the sediments. One cold and snowy, flaky night a couple of nights ago. So we've got some more samples to look at that center of distribution of the CIS.
But no
All right.
alerts on the way down yet.
Okay, that's what we like. Oh, and David Allen actually wrote and thinks it's a Hanasaki, which would even be more stunning. I mean, well, the whole thing is stunning, that crab, but I'm sure you were doing a lot more than that on board. But that stole the show tonight, so that's good. With that, I guess any other questions on phones or texts or anything for Katrin or Lee? David Allen, have at it.
I think you mentioned this earlier on, Gay, or maybe it was Leah or Katrin, but I think it's really important for folks to, or maybe to have Katrin and Lee to sort of reflect on this idea of when and where might people find sort of final results from this cruise? Because I know science moves a lot slower than we might be able to present in this context or in post-field season meetings for IARPIC and other contexts. So yeah, just this is not even preliminary results. This is the big. This is
Yeah.
you're not even
Well,
at port
This is
yet.
the collection.
Yeah.
Yeah, well, we do publish, of course, the papers, and we've been sending out these daily communiques about what we're doing each day on the ship. So I've tried to, for example, the Don Anderson led a paper that was published in the proceedings of the National Academy of Sciences on the harmful algal blooms, and so I just gave the link in that. So we need to keep coming back to Nome or keep providing these great talks, and we'll just get the information out the best we can. But just as an example, giving a link to, well, here if you want to really read the gory details, here's a paper that was published in a very prestigious journal about the HAB issue in the Bering and the Chukchi Seas, so you can get all the details there. And we need to keep doing that, and that's our ultimate product that as scientists is to have our work tested by peer reviewers to say, yeah, is this really good? Is it worth publishing it? And then making that available, and also trying to prevent paywalls from preventing you from accessing it so that it's freely available to everybody.
But equally important is, other venues such as, we do also present at science conferences. The Alaska Marine Science Symposium is typically one where a lot of us go and present, but also understanding that reaches a certain audience and not another audience that is also interested in these results. And, they actually suggested before the cruise that maybe we could do one of these Strait science talks before and one after the cruise, and it was just the timing just didn't quite work out. It was a little too much all at once, but I think it is a good idea to come back with more results and really reporting also to the region where we sample, and not just report to other scientists. So this is a great venue.
I think I would just add, North Pacific Research Board has really recognized this, that it's important that we go back out to these communities and work with them. And so I think, for example, the program they're spinning up to do a comprehensive ecosystem study of the Northern Bering Sea requires us, or the people that are involved in that, to go to the communities. So go visit Gamble and Savunga and Wales and Ref and places, all the communities that have a stake in this, to tell them, well, this is what we're finding. And I know from my own personal experiences going to some of those communities that people there are really interested in what scientists are finding and trying to compare that with their own personal knowledge.
And I would add too, David, that in the Bering Strait region, we are a part of the ecosystem being studied. I mean, we are a part of the ecosystem here, the utilization, comprehensive utilization of the maritime environment for everything. And so I think that what Katrin and Lee, what we try to do with Strait Science is to give an opportunity for this region in particular to hear real time some of what is happening. And I hope you guys come back when you get all your analysis and tell us even more exciting news. Hope it beats the crab. Well, I don't hope it, I hope everything's under control and good is what we're hoping. But seeing a crab at 71, that does open a lot of questions. But we are part of the ecosystem, and communities do need to be getting the data, the information that in your results right away. People here all up and down the coast from Utqiagvik down to Bristol Bay, but especially in the Bering Strait region, we up on north, we are adapting every day, or having to adapt every day to the environment change, the climate change, the biological changes at sea, the industrial changes at sea, the geopolitical changes at sea in this region. And it is pretty comprehensively overwhelming, and it helps every little bit. And of course, the Nome Nugget is on tonight as well, and they will be probably, or not, writing this up. But if that case, if they do, then it's going completely into the 20 tribal, the 16 communities and 20 tribes that we have in the region. So it's really trying to get that data back to the people who need it. I know there's lots of other obligations, but I'm always grateful for researchers coming on and talking to us here in the Bering Strait region when we get questions and things from elsewhere. And if people are not on right now, they will be watching it on the YouTube channel. So thank you both, Katrin and David Allen and Lee Cooper. So we're really grateful. And there is a chat. Now another surge of interest in a deep water port of Nome. Yes, while I don't know if Lee and Katrin, if that happened while you were at sea, but we're moving ahead, and it looks like Nome will start dropping rock and extending the jetty, and start on that deep draft port next summer. So just so you know, so you can tell SIKULIAQ, they tell us it's going to be like Tetris, but the dock will remain open next year. So I don't know how they're going to do it, but it's going to be tricky. But it'll be interesting to see how they figure that out. But we're not closing down. It's my understanding.
Are you still a port commissioner, Gay?
Yes.
Okay, I have some business with you.
All right.
Let's talk.
Bring it on. Bring it on.
Okay.
All right. Thank you, Danielle. And thank you, everybody, for coming. If there's no other questions, Kyle Dillaplane. Good to hear you, Kyle. Thanks for updating us while at sea. Yes, indeed. It's great. And hi to everybody on board, and we'll see you hopefully tomorrow.
Okay. See you. Thank you, Gay.
Thanks,
Yeah.
everybody.
Have a good
Thank
night.
you.